Dear colleagues, ladies and gentlemen, thank you so much for your kind invitation. Being able to send this pre-recorded communication to you really is an opportunity that I didn't want to miss. Let's make the most of it. Today I'd like to share with you some thoughts on the mechanisms of a stabilization of the wrist, a subject that, as you know, has always interested me. As you know, the wrist is not an easy articulation to understand, and much less to treat effectively. It is one of the most complex articulations of the human body. It comprises 15 bones, 24 joint facets, and 24 ligaments. Furthermore, it incorporates a system of 8 pulleys to control the movement arms of 24 tendons that go across the joint. Why do we need such a complicated link between forearm and the hand? I guess that the necessity to have a large mobility on one side and to keep normal anatomical relationships throughout the entire range on the other are all factors that explain why this joint is so complex and yet so interesting. Yes, we need a mobile wrist to place the hand in the best possible position that allows manipulating all sorts of objects with a minimal energy cost. But we also need to ensure that the joint is stable to sustain considerable amount of forces without yielding or suffering injury. Today, I'll share with you what's the current understanding of carpal dynamics. Let's start by the basics. In kinetic terms or dynamic terms, a joint is meant to be stable when it is able to sustain physiologic loads without yielding. When the wrist is loaded, capsular ligaments are the first to react. Some ligaments, however, may have difficulties resisting prolonged tensions, thus the need for muscles to protect them. Yes, we need ligaments and muscles, but we also need close coordination by the sensory motor system, a specialized group of neural elements that use the proprioceptive information provided by the joint to control stability. But let's start from the beginning. What is the real role of ligaments in wrist stabilization? Not long ago, we all tended to believe that ligaments were passive constraints holding bones together. We all took for granted that ligaments followed the principle of tensegrity, according to which any load applied to a bone on a slope will induce a specific displacement of that bone. Constrained by ligaments, that displacement would generate increasing friction between the bone and the inclined surface. Tensegrity shows us that the higher the tension in those ligaments, the more compression will appear within that joint. On theory, the larger the load, the more stable the joint would be, provided that there is no ligament rupture. According to the principle of tensegrity, any ligament rupture should cause unbearable dysfunction. All ligament ruptures should be repaired urgently to avoid carpal collapse. If that was true, how come there are so many scaphoidal ligament ruptures in the aged population that are so well tolerated? Moreover, if stability depended only on ligaments, all gymnasts should have unstable wrists if we consider the stresses that their wrists need to endure. Indeed, ligaments should not be considered like cables. They are certainly stiff, but not that strong. So, to understand the role of ligaments and muscles in the stabilization of the wrist, first we need to know what sort of displacements occur in the carpals in different loading conditions. In the biomechanical laboratory of the university here in Barcelona, we designed a jig that allowed testing fresh cadaver specimen in different wrist positions. 3 different loading conditions were trialed. 1. Axial loading, 2. Distal traction, and 3. Tendon loading. For the first, we applied an external compressive force along the third metacarpal. In the second test, the loading was reversed, inducing distraction along the third metacarpal. And the third loading condition consisted on the simulation of an isometric contraction of muscles by hanging weights proportional to their physiologic cross-sectional area along specific tendons at their insertion sites. This is a schematic representation of the 3 loading conditions analyzed so far. We used the standard motion tracking devices. We observed and qualified what happened in the wrist before and after sectioning specific ligaments. What about in the first test, axial loading? When an axial compressive force was applied to the third metacarpal, the scaphoid consistently flexed and pronated. The triquetrum, by contrast, tended to rotate into extension and supination, just the contrary of what we found for the scaphoid. Consequently, the distal row tended to pronate, the scaphoid to flex, the triquetrum to extend, and the entire carpus tends to slide down the slope of the distal radius. These are the most consistent rotations and translations found in this part of the study. As you can see, the thumb exhibits a forward rotation. A forward rotation meaning that the thumb goes palmally, and that may only appear if the distal row pronates. The ligaments most likely to constrain those attempted displacements were the long radial nail ligament, that prevents ulnar translocation, the palmar lunotrochlear ligament that prevents trochlear extension, the dorsal intercarpal ligament and dorsal scaphoid ligament that avoids excessive flexion and pronation of the scaphoid, and 4. The scaphocapital ligament palmar that controls distal row pronation. As a group, we call that the helical antipronation set of ligaments, HAPLs, and they merge from the volar side of the radial styloid, rotates in a clockwise manner around the central column until it inserts distally on the volar aspect of the capitate. What happened when there was not a compression but a distraction force applied to the third metacarpal? The scaphoid consistently extended, whereas the triquetrum into flexion, just the contrary of what we found for the axial compressive load. In that case, the wrist was dragged towards the radial side, the scaphoid was extended, the triquetrum flexed, and the distal row rotated into supination. In this particular loading condition, 2 groups of ligaments constrained in a counterclock direction all those rotations. We call them helical anti-supination ligaments, and they are arranged like a helix around the central column of the wrist. One protects the radial corner of the wrist, the other protects the ulnar corner of the wrist. From all those observations, an important concept arises. Ligaments do not like to work alone. They cooperate with other capsular ligaments, trying to protect themselves against excessive tension of their fibers. Yes, it's a teamwork type of job, the one of wrist that the wrist ligaments have. But let's move ahead and see what's the role of proprioception. The first concept we need to emphasize is that ligaments are complex structures containing sensory receptors aimed at providing proprioceptive information to the sensory motor system to ensure carpal stability. When there is excessive tension in a ligament, some receptors contained in that ligament will generate warning messages to the central nervous system. Based on this information, the sensory motor system decides which muscles need to contract and which need to relax to prevent further ligament damage. Obviously, the system is designed to protect the ligament against excessive traction. Should the muscle reaction be too slow, the ligament may be at risk of rupture. In 2007, our good friend Elizabeth Haggard from Stockholm in Sweden, which you know as one of the pioneers of this type of research, she used immunohistochemical analysis to demonstrate that ligaments don't have the same amount of receptions. From a very densely innervated ligaments to ligaments with almost no innervation, a wide spectrum of possibilities exist. For the first time ever, she provided evidence of the existence of wrist ligament muscular reflexes, with early reactions being interpreted as a joint protective function and later contractions indicating supraspinal control of wrist stability. Interestingly enough, activity changes were not restricted to one muscle for each wrist position. Combined reactions were the rule, and all reactions were in terms of muscle contraction. Sometimes there were negative reactions as well, that is, muscle inhibition. So, what's the real role of muscles? Why some muscles are asked to contract while others are told to relax? Moreover, how could muscles prevent carpal collapse if their contraction is likely to compress the carpus? To understand and decide on these questions, we decided to investigate this further. Now we wanted to know what displacements occur in the carpus when some individual muscles were isometrically loaded in a predetermined role. What we found was that some muscles induce pronation of the distal row relative to the forearm, while other muscles induce supination relative to the forearm as well. So how can this be explained? To get an answer, we placed metal markers within the substance of 5 tendons. The markers were set proximal to the wrist, and here in yellow, and at their distal insertion site in blue. Then we finally cut the specimen to get all markers in one axial X-ray view. The experiment demonstration was that all tendons change direction at the level of the radius. The issue, for instance, is dorsally located at the level of the ulna. By contrast, it inserts onto the medial corner of the 5th metacarpal distally. Because of this change in direction, contraction of the issue is likely to generate a pronation moment to the distal row. By contrast, there are other tendons which have opposite oblique nurses at the level of the wrist, inducing a supination moment to the distal row. That was the key to understand how muscles contribute to carpal stability. Muscles are like reins of a horse. You may pronate or supinate the distal row relative to the radius depending on what rein you use. If you pull left, in this case, if you pull extensor capiloradialis longus and abductor pollicis longus, ECRL and APL, the horse will turn left, that is, in supination. If you pull right, in this case the ECU, extensor capio ulnaris, the horse will turn right to pronate to the distal row. If your carpal is unstable and your scaphoid is about to collapse into flexion and pronation, you need to contract only the muscles that will supinate and extend the scaphoid. By contrast, if your carpal is unstable and the issue is only pulled, you will protect the ulnar column of the carpal. Well, these are the actual figures of the effects that we calculated produced by isometrically loading each of the 5 varus motor tendons in 30 cadaver arms, fresh cadaver arms. The distal row will rotate into pronation essentially by acting on the ECU or into supination by pulling the ACRL and APL. It's no wonder then that the ECU was found to be inhibited when the scapholonate interval was at risk. The ECU's strong distal row pronator, as you know, that's a potential enemy, very unfriendly with the scapholonate instability. You may see in the instant shots of a video taken after sectioning the scapholonate ligament and running the same experiment. Only one muscle increases the scapholonate gap to the right, the ECU. All other tendons have a supination effect closing the gap to the left. What's the mechanism for the ECU to increase that much scapholonate gap? Well, you know that by permeating the capitate, you are inducing tension in the scaphocapital ligament, and this would result in a scaphoid pronation and an increased tension to the dorsal ligament. Indeed, if you have a partial tear of the scapholonate ligament, a distal row pronation could increase the scapholonate gap and make the instability much worse. In summary, when there is excessive tension in a ligament and the ligament is reaching its yielding point, the mechanoreceptors contained in that ligament will generate a warning message to the central nervous system. Based on this information, the sensory motors system will decide which muscles need to contract and which ones need to be inhibited to prevent further ligament damage. So, if the wrist is exposed to a supination stress, the pronator muscles, that is the ECU, will be there to avoid further injury. By contrast, if there is a pronation stress, the supinator muscles, essentially the ACRL and APL, will be called in. At this point, I would like to dedicate this work to the memory of the late Professor Josep Maria Domènech Mateu, whose presence in the anatomy lab still is painfully missed. I want to acknowledge all my pals in Barcelona for their patience, for their hard work and enthusiasm of the work we started long ago. Thank you very much. Thank you. Bye.